Hello.
Hello, Dr. Huntsman. How are you?
Oh, very well. Yeah, looking forward to today's rounds.
Yeah, Dr. Livu is going to present today on our RNA, and next week, Dr. Kote and Dr. Mike Nemo, they are presenting on scientific aspect of department round, strategic plan.
Sorry. So there's I didn't catch you. Sorry. What are they presenting on?
On the strategic plan,
So
strategic plan,
so for the pathology department strategic plan.
yeah, as a scientific research aspect, I guess.
Okay, fantastic. And good. And I saw that you sent me a little bio here.
Yeah.
Hello.
Hello.
Hi, Lai. So thanks so much for doing rounds today.
Oh, yeah, of course. My pleasure.
I'm personally very excited by these rounds as a learning experience for me, but I'm sure others will benefit too.
I hope I'm not sure about the audience, so I kind of have a range of things. So yeah, some part might be enough and some part might be a little bit more advanced, but I feel like maybe that's good to just have an introduction for the field. So
yeah, because a lot of people like trained a long time ago, and
right.
field has moved
Yeah.
quite substantially. Fantastic. So yeah, so we're what we tend to do is you've got a bit of time here.
Right.
So I don't know if you want to get yourself some tea or anything, or
maybe I would like to try to just share and then make sure that everything is good.
Absolutely.
And you see my screen or my presenter screen?
No, we're seeing actually your screen. So yeah, it's good to
Okay, so I'll swap it. It's good now?
Brilliant.
Okay, yeah. So because there was some like update with Zoom and then I have some, so I just want to make sure. Okay, it's good, right?
Looks good, and I can hear you very clearly.
That's great. Okay. So I think that should be good. Then I'll stop and then we'll share later. Okay, great, awesome.
Okay, I'm just going to make tea, so I'm going to turn off my camera for a second, and I'll be
back. Great. Yeah.
Okay, so I think we can start inviting people in now. This is good. Okay, so welcome to province-wide rounds. I know there's still more people coming in right now, but I'll start off by announcing next week's presentation. Doctors Cote and Nemo will be describing elements of our department's new strategic plan, which I think should be broadly engaging and interesting for all department members. Today, we have a special guest, Dr. Lai Vu, who's a CRC Tier 2 chair, recently recruited into the Terry Fox Laboratories and the Faculty of Pharmaceutical Sciences at the University of British Columbia. She originally did her bachelor's degree in that field, and with a PhD and postdoctoral training in Memorial Sloan Kettering, where she worked on RNA binding proteins, RNA methylation, and the pathogenesis of myeloid leukemia. Since her arrival here, she's won multiple awards, most recently one of the V Foundation scholarships, which are incredibly prestigious, and is already in a short time has become a very valued member of our community. When I did my training, there was this linear story of DNA to RNA to protein, with nothing in the spaces in between and no back chat allowed. And things have become much more interesting and more complicated. And I knew I would benefit from a talk on what's at the forefront of RNA biology research. It's got a cancer theme, of course, with a focus on myeloid leukemia. But I was really excited that Professor Vu is willing to speak with us today. And there'll be some time at the end of the presentation for questions. So please grab control of the screen, and I'm looking forward very much to your presentation.
Thank you, David, for such a kind introduction, and I'm very excited to be here and share with you, some of the kind of like the topic that very close and dear to me and my lab, and the focus of really the focus of my lab. And I launched my independent research group, and as I mentioned to David earlier, it's like I'm not sure about the audience, so you will see it's a range of touching on something more basic, and toward the end I want to kind of introduce a little bit of the kind of state of the field, and very much welcome questions and interaction in the future to putting this context more into clinic and to understanding the pathogenesis of the disease. So with that, today I'll focus on the RNA modifications in myeloid leukemia, but we'll start with some of the more overview of these gene expression control, and from there bringing the context of the disease and what kind of the experimental approach and tool that we have in our hand to really investigating this field. So to start, we probably all know that cancer is characterized by multiple different hallmarks, including these increase in proliferative potential or resisting cell death, and you become immortal and can invade distal and foreign tissue. But underlining all of these very diverse hallmarks is actually a change in gene expression. So either from mutations or transcriptional control, epigenetic, etc. It's all come down to this level to drive the gain of characteristic in tumor cell. So when you look at the change in gene expression, when we study the gene expression pathway, you start from like a gene or the genome, and then really the phenotype in patients or in our experimental model, it's going through actually multiple layer of step, going from, as David mentioned, genome, DNA to mRNA, which we kind of look at here, transcription regulation, and look at, just the phenotype. But in between, you can see that RNA have its own kind of layer of regulation, as well as after the protein synthesis of making a protein, as you all know, they also have post-translational regulation. So the control is way more complicated and multi-layer, in this nature. So looking at the understanding of how these gene expression programs usually deregulated in cancer, a lot of attention have been focusing on characterize the genetic alteration. So the profiling of patient sample and finding out genetic mutations, as well as like we define driver and passenger mutations, and as well as the transcriptional profiling and understanding also a little bit of the epigenetic contributions into modulating the transcriptional process had been the focus in the field. So I think in this step, even though there's still a lot more to look at, we do gain substantial understanding here, as well as the effort to really profiling protein expressions in tumor sample, give us an idea of what protein that we can find and what kind of function that it can modulating to cause these malignant transformation. However, these steps here, when you look from the mRNA to the protein, it's actually a hole in our understanding, and much less has been understand. And luckily, in the last couple of years, the field has been receiving more interest. It is mainly due to the realization that in a lot of study, when we look at the transcriptome and the proteome, we see that they actually not always completely correlate. So you might have an mRNA expressed, but then maybe the protein is not at the same level. In addition, there's realization that post-transcriptional control actually provide a lot of diversity for our proteome. So if you can recall some of these knowledge in the splicing and generations of different protein isoform, and different protein isoform can actually play different functions in the cell at different conditions. And that it also gives the cell ability to quickly respond to certain stress signal from the environment. Instead of rewiring the whole transcription control, now they can maybe modulate somehow the mRNA expressions, or slow down or speed up productions of protein. So it gives the cell more kind of nimble mechanism to respond to those. In addition, there's also realization that there is more interaction between each of these regulatory steps, enable a more complex regulation. So for the people who don't really think like me about post-transcriptional regulation all the time, I want to put this slide to kind of remind everyone about different layer of regulation and mechanism that can involve in this post-transcriptional regulation, which is actually quite complicated. So when the mRNA is first transcribed in the nuclear, before going out to the cytoplasmic, where we all think that it's going to get made by binding to ribosome, and the protein is translated here, it actually undergo many processing steps. So very characterized process of capping at the 5' end of the mRNA, as well as additions of the poly A tail length to protect the mRNA. But the mRNA also can be edited changing the exact nucleotides in the sequence, as well as modify. And before they go out to the cytoplasmic, they undergo splicing. So then you have the mature mRNA with the exon in here and gets exported. So the export process is also very well regulated. And besides getting translated to making protein, mRNA can also be recruited to different granules structure in the cytoplasmic to undergo either translation repression, and at the end of its life cycle, it will get degrade under different degradation program. So any mRNA at the end will be, undergo decay and get rid of in the cell. So that's the endpoint of their RNA life cycle. In addition, there's a multiple evidence that coming from different study suggesting the very deep involvement of post-transcriptional control in cancer and tumorigenesis. So there's a lot of mutation actually by identifying the RNA processing component, in particular RNA splicing machinery. There are also a lot of aberrant expression have been found in this RNA binding protein that really modulating a lot of the process during the regulation step. And the translational machinery itself actually got hijacked in cancer cell by several different signaling pathway. Some of the famous one, including like the RAS or PI3K AKT pathway. And the genetic evidence come from these identification of this increased predisposition to leukemia development in the ribosomopathy disease when there's a hereditary disorder in mutation of ribosomal subunits and deregulation of translation control that result in high propensity for leukemia development. So all of these evidence really suggesting, there's an important role of these regulation process and their opportunity to study them and then nominating, new therapeutic target. Another very important factor that really allowing the field to arrive at this state is that the availability of enabling technology in the last, I could say, 10 years. So before we probably only can see how mRNA, the abundance of mRNA as well as the protein, but couldn't look at anything in between. But now with these technology that allowing us to, map the interactions of protein and mRNA, as well as looking directly at the modifications of RNA, not just a naked molecule, as well as assay for the activity of ribosome that bind to the mRNA and controlling the translations efficiency as well as translation rate of making protein really enable us to investigate and gain insight into this process. So with that interest and the available tool, it's become important to really answering the question of, how the deregulated RNA metabolism can contribute to the cancer phenotype. And then by understanding the mechanism and how it works, maybe we can in the future therapeutically, target it for the better treatment. So as I show in that general slide, you can see that a lot of protein actually interacting with RNA at different state to modulate the regulation. So in that kind of regulatory network, there are two components. So it's a protein that regulating it. In this case, they are RNA binding protein. And another one, of course, is the mRNA molecule itself. So for this talk, I won't be touching a lot on this RNA binding protein, but I want to really highlight them as a key controller of many process involving in this layer of regulation. And RNA binding protein actually in human, there's now over 2,000 of them have been identified. And as you can see here, they involve in multiple process to mediate both transcription as well as post-transcriptional control and translation. So studying them is one of the very important area in the field. And then I want to then move to the RNA molecule, which is another component of these network. And if we used to look at the messenger RNA, which we look at all the time, and actually I don't have it here, it's a kind of a boring one string of molecule, but actually RNA is quite diverse. So beside messenger RNA or mRNA, we have ribosomal RNA, which is the most abundant RNA in the cell actually. And we also have transfer RNA, which is tRNA here, that we all know involved directly in protein synthesis and translation by bringing and then decoding the genetic codes on the mRNA. But at the same time, there is a big group of RNA that now really coming into the field is a non-coding RNA that including both long non-coding RNA, so they not like they characterized by longer than 200 nucleotide, and as you can see, they can form different structure and also interacting with protein, as well as small non-coding RNA. And one group that people might be more familiar with is microRNA, but also there's a group of a small a nuclear RNA, which is a SNR RNA, play a different role in both mediating the maturations of mRNA and contributing to the rRNA synthesis as well. So here I want to highlight the diversity of RNA as well as the different structure that they can adopt. And if you can see here as well, there's a note on different modification that you can find in the RNA. So in fact, all of this RNA is heavily modified, especially the more structural RNA, including rRNA and tRNA. So RNA modification is known for a long time, but in general, they would be considered as the irreversible decoration on the RNA structure, that making them have maybe increased ability or enable some of the catalytic function. But what kind of make the field more excited recently is that the realization that some of these modifications that actually are reversible, that indicating that they might be dynamically regulated, and with the regulatory potential, there might be potential to actually manipulate and interfere with their function. So with the modification in all the RNA, there actually over 170 modifications have been identified so far. And as on this graph here, you can see that the modification can actually be found in all four different nucleotides. But most of them is found in RNA and tRNA, and some is found in messenger RNA. And the one that kind of circle and highlight here is found in mRNA. So even though the most abundant modifications is in RNA and tRNA, the focus of my talk today actually is on the messenger RNA and these different more reversible and regulatory modifications in the messenger RNA. So with that, the finding that the mRNA is modified not only at the 5' and also the 3', a very classical modification, but also internally by different modification, really push the idea that there is the layer of control, and there's a term that we'll coin in the field, which I would say is still controversial, but at least kind of reflects some of the thinking on maybe the mirroring the concept of the epigenetic field that called the epitranscriptomic, which means that it's not changing the exact sequence of the mRNA, but instead changing the modifications on it. So the epi part. And if you're looking on this graph, there's one modification that actually the most prevalent internal post-transcriptional modification, which is the called the M6A, which is the RNA methylations in the 6 nucleotide. So the RN6 methyl adenosine, which is M6A, is actually a very dynamic modification. So very similar to the epigenetic control that you might more be familiar with. So this one also get deposited by a group of proteins, and here the metal 3 is an enzymatic kind of enzymatic subunit called the writer. So there's a depositions of these metal mark in the location. And the modification can also be removed by the eraser protein. And here listing out the FTO and ALK-BH5. And interestingly, there's known that there is different protein that can be designated as reader protein that basically recognizes modification and drive the downstream consequences. So for the last 5 years, the field had now gained substantial knowledge of how these modifications work and what is the consequences of modifications within the mRNA molecule. So as you can see here, the modification is deposited co-transcriptionally. Oh, I apologize. This happened before. I test today, it didn't happen, but okay. I'll restart again. My Zoom crashed some point. It's
our building is notorious for.
yeah, it's like, and I think it's just the update on their security or something. So good.
No, not yet, actually.
Oh no.
Nope.
Oh, let me
To share screen.
I need to share the screen.
Brilliant. We're right back where we started.
Okay.
Fantastic.
I hope it will stay until the end. Okay. So I was saying that the M6A deposit co-transcriptionally, and when it's deposit, it can be recognized here by reader proteins, even in the nuclear, and that can regulating the splicing of the mRNA. It's also play role in modulating the export of the mRNA from the nuclear to the cytoplasmic, where when it's outside, it gets read by other additional reader protein that mediated either mRNA degradations or translations activity. So a very important question in the field when we looking at this is very general, is where and actually on which transcript do we find M6A? Are they exist in every single transcript, or there's a group that is specifically enriched for it? So this question is actually very important, and the ability to mapping it really make the field moving forward significantly. So we know that with the transcriptomic profiling, the RNA sequencing is a method of choice. So here, just to kind of walk us through, when we have the sample of interest, the total RNA can be extracted and enriched and subject for sequencing, when we then can map back to the genome and determine the differential expressions of specific gene, call for variation, discover a novel transcript, and even look at RNA editing. But for the modification, because we cannot get the direct information from here with just a transcript, so we always need to have an assay that either enrich for the modifications or make a mark for the modification, so we can distinguish between the modified transcript versus the native transcript. So with that idea, the first approach in the field was using the antibody that now can help enrich for modified transcript. And in this assay, you have the antibody that specifically recognize M6A mRNA. When you pull down and then now subject these pull down mRNA for sequencing, we have enriched for what RNA have the modification instead of just profiling the whole transcriptome. So later on, the field developed another kind of upgraded version of this technique, when now using the specific antibody-induced signature, mutational signature by protease degradations of UV cross-link antibody to the mRNA molecule. The profiling can be done at the single nucleotide resolution. So we can pinpoint exactly what A within the whole transcript there is the modification. So using this two technique, we gain a substantial understanding of where the methylations is found and how many transcript is actually there. So several profiling has done in both mouse and human and various tissue, found that's about 25 to 60% of the whole mRNA of the transcript is actually methylated. And there's a distribution of where the M6A methylation can be found. So it can be found both substantially in the coding region, also quite a lot in the 3' UTR, a little bit in the 5' intronics, and also intergenic region. And within a certain transcript, there also the specific profile of enrichment that you can see where the modification is at. So very low in 5' UTR, kind of distribute across the coding region, enrich toward the end of the coding region, right at the beginning of the 3' UTR, and then reducing here. So this signature are actually true for a lot of different assay as well. And also by looking at where the modification at, there's a consensus motif have been identified called a drag motif. When there is a conserved AC well, next to the A when it's modified, you always find a C, and then the variable of where you can find different nucleotides along that motif. But that's really how the field moving forward, knowing where it's at, and then now can look more in the biological process that is influenced by the modification. So then a landmark paper that really also push the field moving forward and then pick a lot of interest is the study done in 2015, when they first found that the M6A RNA methylation actually important to facilitate the resolution of the native pluripotency toward differentiation. So here, using the mouse embryonic stem cell, the author found that in the very native state, like super stem state, there's a lot of mRNA that's responsible for maintaining these native state. It's has to be methylated when they undergo from the naive to the prime state. So with this methylated mRNA, they would be degraded, so allowing the cell to give up their native state and become prime for differentiation. So when the method 3, which is the writer of these modification, is removed, there's no result. Now there's no depositions of this methylation. Now all the mRNA sticking along, then the cell cannot resolve their native state and will stay here and have impaired in differentiation. So this study really key and suggesting that the methylation can play a direct role in regulating stem cell function. So when that first study came out, we're really interested in knowing, okay, so if that is important for stem cell, how about its role in cancer? So this is the work that I done during my postdoctoral study, and this really when the field doesn't have any idea yet of what it does. So for us, when we start asking this question, we access the TCGA Cancer Genome Atlas database and look at the method 3, which is the writer and the enzymatic subunits of the writer complex across different cancer type. And what we found is that actually the method 3 is expressed the highest in acute myeloid leukemia, and it's actually the disease that we study. So we really feel lucky to have this observation. So just a quick background on the acute myeloid leukemia and what the disease is. So the myeloid leukemia is the disease where the dysfunctions of blood production is characterized by a blocked in differentiations of a stem and progenitor cell to make the functional and differentiated cell in our blood system. And then also there's a gain in cell renewal of the progenitor downstream hitting this population that allow for the transformations of normal stem and progenitor cell and become leukemia initiating or stem cell. And these cells have this self-renewal capacity as well as therapy resistant, and it's postulate to be the one that drive relapse in patient and then regenerate the heterogeneous leukemia blasts that we found in patient, where we characterize them by increased proliferation, increased survival, there's impaired differentiation, and the sustained cell renewal. So my lab in particular very interested in understanding these characteristic of the cell, because they in clinic, when we know that the overall survival of myeloid patient, leukemia patient is not great, the relapse really devastating and result in much poorer prognosis and outcome in patient. And we think the leukemia stem cell with that feature is really the one that driving these phenotype. So with that interest and the observation that MELA3 is highly expressed in AML, we then decide to look further in how the MELA3 functions in leukemia cell. So the first thing we did is we checked for MELA3 expression across different AML cell line in the lab compared to the normal stem and progenitor control, as well as the expressions in primary patient sample. And as you can appreciate here, we see that MELA3 expression is highly upregulated in both cell line and primary patient. We're also able to measure the M6A level in the total RNA from the cell and see that compared to the normal, the leukemia cell also have more M6A RNA as well as in the primary patient. So it's correlate very well with the expression level of the writer enzyme. We then next ask what would happen if we take away the writer, which means reducing the methylations in the cell. So we found that when we knock down METO3, the cell have very significant inhibition of growth. We see very significant inductions of differentiation in the leukemia cell, as well as inductions of apoptosis. So these phenotype translated to the increased survival of recipient mice when METO3 is depleted. So with this, we think that METO3 is really important to maintaining the leukemogenesis. So to further understand how it works, we decide to map the RNA methylations target using this MyClip assay that I described earlier. And on top of that, to really kind of layer up all the level of regulations, we also perform RNA sequencing to look at the transcriptomic profile in the cell. We also perform ribosome profiling, which mapping the ribosome binding to mRNA. From there, you can infer the translational rate of an mRNA, as well as looking directly at some of these expression at the protein level, which is the functional level of any gene by the reverse phase protein lysate microarray. The idea is when we can layer all of them up, we can figure out exactly what's the consequences of methylations and loss of methylation. So with the MyClip assay, we can map and gain the exact distributions of the M6A as we expected. And in the knockdown of METO3, when we perform transcriptomic profiling and then bin them up like to the M6A modified and non-M6A modified, what you can appreciate here, there's a shift in the curve of the non-modified M6A to the left. And then the M6A is to the right. This is indicating that the M6A marked transcript actually have higher mRNA abundance when METO3 is knocked down. So suggesting that when they have M6A methylation, the mRNA tend to be degraded. On the other hand, when we look at the RiboSeq experiment, when there's a translation efficiency when METO3 is knocked down, you can see that the curve of the M6A, the blue for the M6A modifying transcript actually shift now to the left. And that's indicating that M6A marked transcript actually have a lower translation efficiency when METO3 is knocked down. So here, I hope you appreciate actually there's a difference between the response at the transcription level and the translation level. And we look at the further into a subset of embryonic stem cell actually enriched in the M6A direct transcript. And we look at the RNA sequencing in RiboSeq. You can see also here a flip. So with the RNA sequencing, this gene set is actually downregulated, upregulated. But with the ribosome sequencing level, they actually now negatively enriched, which is downregulated. So there's a flip in the RNA and protein expression. We also able to map the M6A target in the primary patients, and very similar to what we saw in the leukemia cell line. So we also have information on the protein and also different signaling pathway downstream by looking at the protein array. And when we run the expressions when METO3 is knocked down, we've seen several interesting target popping up. So there is including phosphorylation of Richter, GSK, AKT, as well as BIM or Cas3 apoptotic proteins. And when we're now looking at the target gene that was identified as the M6A target, we found some very prominent target, including PTEN, BCL2, and c-MYC. So MYC is one of the most important oncogene in a lot of different cancer. BCL2 play a role in apoptotic mediate, and PTEN in cell proliferations and differentiation. So we decided to look further into this three gene, and very similar to the trend we actually observed in our sequencing. So here at the mRNA level, we can see that after knockdown of METO3, we have a lot of mRNA of c-MYC, BCL2, and PTEN. So they clearly upregulated. However, if we look at the protein by Western blot, the c-MYC, BCL2, and PTEN, all three of them, the protein is markedly downregulated. So again, this mRNA and protein is not correlated. And without META3, we actually lose the protein expressions of these three important targets. So with that characterization, we then decide to look maybe on the potential therapeutic interference. So we all know that PTEN is actually a negative regulator of the PI3K kinase pathway with activation of AKT, which then result in increased survival and reduced differentiations in leukemia cell. So we asked if we now take away PTEN or like in our system with AKT is activated, can we eliminate or like reverse the phenotype by now inhibiting this pathway? So that's exactly what we did. So we treat the cell with inhibitor of PI3K as well as AKT, and at least in the differentiations phenotype, we're able to partially rescue the inductions of differentiations when META3 is knocked down by putting in the inhibitor of these two steps of the pathway. So from that study, we was able to demonstrate that META3 control the translations and mediated differentiation of both a normal and a malignant hematopoietic cell. That is really important to contribute to the leukemogenesis both in vitro and in vivo. So right after our study, there's a number of studies actually came out right after showing the similar phenotype when they manipulated META3, as well as showing the involvement of other proteins of the pathway, including the FTO, which is the eraser protein, in myeloid leukemia. And since 2017, only after, I would say, 4 years, we're very pleased that the field made a very significant milestone that they screen out and identify small molecule inhibition of META3 as a strategy to eliminate leukemia. So this is a structure of these first generation of this compound, which I believe the company have further improved. And they found that they can inhibit the activity of these enzymes in the very low micromolar to the nanomolar range, and demonstrate some of these efficacy in animal model. So here showing with the luciferase with vehicle and then treatment using two different AML model, that there is reduced in tumor burdens when they treat with META3 inhibitor, and that result in delay in increased survival of recipients animal. And with this result, actually in 2022, there's a clinical trial have started by the company called Storm, testing that exact molecule, but I think it's a different version, improved. And they have enrolled patients and expected to actually report preliminary data by the end of this year. So I think this is particularly very exciting because what we found, a couple of years ago now can actually be translated. And here showing that what we see in AML, actually some can be recapitulated in different hematologic malignancy, ALL, and a lot of different mechanisms have been figured out. But one here also want to highlight is then after the development of META3 inhibitor, there's also inhibitor that develop for other regulatory proteins in the pathway, including both the eraser and the reader. So here are different compounds that actually in various states of development to inhibiting and manipulating this pathway, which is very exciting. So this makes us look like a lot have been figured out, and almost is that any new thing that we can further look? And I would say yes. So in the field now, the key remaining question that we really asking ourselves is, as I describing to you, the methylation actually quite conserved and is universal. So it's found in mammalian cell and human cell, and it also found in yeast. But there is definitely a context dependent. There are some subset of target is methylated in one condition versus other condition. And we know that they are dynamically regulated during normal developments and disease. So how actually that work? So there are a focus in the field looking at maybe modulating activity of the protein that involved, so involving in like maybe post-translational modifications of those protein, and then control where they be localized in the cell and then mediate its function. There's also idea that they would methylations modification not acting alone. It would have to kind of interact with other process of RNA processing, including transcription, splicing, decay, and translation. And one important part is the context-specific M6A target. So can we identify very specific target in different tissue, in different contexts, and then whether we can really map down what is the target that is functionally relevant, and what is the selectivity there. Another aspect that I kind of want to introduce to make it even more complex is if we think about DNA modifications, we would think with the DNA, you have two allele, right? So either you would have both allele methylated, or one methylated, or no methylation. So it's a bit of these binary, like a three tier. But if you look at the mRNA, they exist in a range. So we're really interested in understanding the stoichiometry of the M6A modification, whether they are binary, as we think, in with the DNA, or they actually exist in a gradient. And then in that case, the frequency of these M6A modified transcript versus a non-modified transcript might have a significant biological differences. So with the, we know what transcript it in, where it is, but this we don't know a lot. And we have to also come to appreciation of these heterogeneity among different cancer population. With the single cell sequencing, now we know that the previously thought, very homogeneous cancer cell, actually very heterogeneous. So different state and different, cell state. So it become important to ask these questions in the context of modification. So I went through these two assays, the MIRIP and then the MICLIP, which is the MIRIP using the antibody. It's the most popular assay in the field. It's because it's very easy and very well established. So any lab can actually performing it, and they can use low to high input RNA to run the assay. However, there's so many different, so many cons in the assay. One is that it's antibody dependent. So by itself, it's very promiscuous. So it can bind non-specific for different another modification as well as different site. And then also there's coverage issue if there is RNA structure that doesn't allow the antibody to come and recognize. They have very limited resolution. So you have a broad peak, so you don't know exactly where the methylation A is. And there's no information on the stoichiometry. So the MICLIP solves some of that problem with the single cell nucleotide. So you know the A site, but there's still limited quantification, as well as still antibody dependent, and by itself is very complex and difficult to perform. So the kind of commonly used technique cannot solve what we want to solve. And we put out an ideal technique would be to have very high resolution. So you won't capture every single transcript, every single site, will be high throughput and easy to perform, would definitely need to give the stoichiometry measurements. And ideally, as I mentioned, if we want to correlate the methylation with other feature of RNA and other regulatory process, it would be great to simultaneously capture these feature. And for a lot of application in stem cell or clinical, ideally it will work with very low input with high fidelity. So I would say we don't have yet the ideal technique, but there's a couple that coming out very recently that bring us pretty close to it. So this assay actually just came out this year in 2022, is allowing the genome-wide mapping of RNA methylation, and it's not dependent on the antibody and pull down. So in this assay, what happened is let's take advantage of an enzyme called TAT that can actually convert all the adenosine to inosines in the mRNA. So if you imagine if now you can treat the mRNA, you can convert A to I, but the A that have methylation will be protected by these conversion. So now when we sequence with the conversion, A go to G in our sequencing, but then with the methylation, the A is conserved. So by comparing now the ratio of conversion to G versus the native A, which inferring the RNA methylation, we can have the percentage of modification mapped down here. So I hope you can kind of appreciate by the signal showing here. So using this technique, we can also now do the single nuclear resolution because we have that resolution in sequencing. It's antibody independent, but for sure. And then the ability to comparing, from the G and A ratio give us the quantitative stoichiometry. And we actually can work with the low input capacity, which is really good. The only disadvantage of these assays is that we have not been able to adapt it at the single cell level, so we don't have these heterogeneity assessment. So another assay that also developed quite recently, the first one in 2019, and this one actually last year, that using a different approach. So as you might recall, the methylation is read by a reader protein, and there is a domain in the protein called YTH. It's the canonical reader domain. So in a lot of technology recently developed, we can make fusion protein. And in this case, with this reader domain, we can fuse it to an enzyme called AppleBack. So AppleBack is an RNA editing enzyme. As I mentioned, you can actually convert it, different nucleotide here to by edit it. So in this case, the A can be edited to use. So with the A agent to C to T signature, you can actually measure the level of M6A by frequency of editing of C to T in the A and C that next to it. So using this technique, you can transduce cell, isolated RNA, or taking cell directly into the single cell RNA sequencing platform, and then perform profiling of M6A. So with this method, we can have a near single nucleotide resolution. It's again, no antibody, so we get rid of that problem. They give certain quantitative stoichiometry because we can compare between the ratio of editing. It's very cool that it's actually enable us the single cell capacity. However, there is some limit to the YTH binding and then requires some manipulation of cells, so it might not be applicable for a primary clinical sample. And the last one, it's not there yet, but I kind of want to just introduce to see where the field is going, is now with the use of the nanopore native RNA sequencing. So previously, we know that it's a short read Illumina. So in this one, the difference is the ability of having the direct mRNA going into the equipment, which set up by here, actually is a pore. When the molecule passing through, each of the nucleotide will give an electrical signal. And depending on their signature that is showing here, you can actually decipher whether it's a T, G, or A, and this allowing for the sequencing of a very long, a much longer transcript, as well as the detections of modification when there is an introduction or like an error in the electronic current. So one disadvantage of this assay is that they still require quite high input, but I think it's getting there, like reductions of the amount of input. If we can go with lower input, I think that will be a very promising approach in the future. And the most advantage here is the ability to then integrate analysis of many RNA feature because we capture all at once. And just to highlight, we study the modification, and then now with the tool of CRISPR-Cas9 and CRISPR-Cas13, we also have the power to actually come in and directly manipulate the modifications using this technology. So we just not profiling it and see where it's at. We can actually putting in and removing it, and then ask the question of what is the biological significance of these modifications. So I will then end with this slide, really showing that what we study so far is in AML and in blood system, but a lot of these concepts and a lot of these findings actually translated to many other cancer type, to breast cancer, brain, lung, hepatocyte, pancreatic, and then bladder cancer. So I really expect, a lot more insight will be gained, and hopefully what we continue doing in the blood field will be beneficial for everybody else in the solid tumor and other system as well. And with that, I'll end my talk, and thank you for your attention. Happy to take any questions.
Well, thank you so much. That was a brilliant presentation. And the data you showed in the middle of your presentation on how poorly RNA levels correlated with protein levels in your experimental system was sort of terrifying, really.
Yeah.
Because it suggests that a lot of conclusions which will be made by myself and I guess many others in many papers are just flat out wrong. So let's open this up for questions. You could either raise your hand electronically or bark out questions.
I would just have the comment there. I would say in general, the mRNA should reflect quite well. It's just in the manipulations of a player that involving input transcriptional regulation, mRNA would not be enough. So yeah, I think that we see a very strong response because we actually manipulated META3, which regulating this whole process. And completely honest, when we see the result, we was like, our riboseq was wrong. We were like scared. But when we run the Western blot, it was like, oh, actually the protein is what it is. So we feel more comfortable. And then more people actually also come out showing in different gene and protein the similar phenomena. So I think the more I think I'm in the field, I feel better about that result.
Yeah, are there, when you think about different cancer types where RNA modifications, particularly the M6A methylation, may be important. Would they be, would more primitive cancers, so cancers which are more embryonic in nature, or cancers which reflect more mature adult tissues, where would you go in the solid tumor space to study this?
The thing is, and in that last slide that I show, almost now, every cancer type, they found that it's relevant. So from the embryonic study at the beginning, we would think, right, like something that more kind of embryonic in nature would be more important. But it seemed across, and I think the important part was that study, they look at the very small window from the resolvement of the naive to the prime state. But once the cell reached the prime state, and now they differentiated, META3 actually need to be downregulated. That's what we found in our system. So the more like mature and adult, they don't like to keep that methylation for too long. It needs to be actually reduced. So I think that's why the context, of when the window of activity is really critical. So when we the follow-up, one of the follow-up study that we did from the leukemia is we make a conditional knockout mice, and what we found is there is the unresolved progenitor state that the cell cannot go through because they lose the META3. So they like the confused progenitor. They can't, they pass the state if they can't differentiate it in the adult. So it's very interesting, where the window of action is at.
Okay, and are there, as we've been studying DICER-associated cancers, whether
right.
it's regulation defects. Are there any particular RNA modifications which change the effectiveness of microRNAs?
Yeah, so that's actually the most asked question whenever I present. So we actually don't see any particular correlations with the microRNA binding. So far the most correlations with the methylation had been the splicing process. So there is the kind of mutual regulation between M6A and splicing. So like, if you have methylation, people believe that splicing be negatively, regulated. But also their studies show now that with the presence of the splicing machinery, you cannot deposit methylation. So that is where it's at. So I don't know whether, modification, let's say, on the pre-microRNA will affect any of these processing, right, step, and then influence the abundance of microRNA. I don't think, on top of my head, there's any study like that. But I think that would actually be a good place to look. Yeah.
Okay, well, thank you very much for your presentation.
Yeah.
I'm not sure if there's any, I'll just look in the chat just in case anything's going on there. No, other than a compliment to your excellent talk. But that was
thank you.
Very, it's like a field where if you close your eyes for 20 minutes, stuff has happened.
Yeah.
And if you close your eyes for a few years, like I seem to have done, the field has completely changed. So thank you for getting us up to speed.
Yeah, and I'm very happy to know if anybody have any question after that or just discuss about the field, feel free to reach out and, we can discuss further. I definitely would like to bring this concept and, the field more forefront of, maybe clinical practice and then think about in like a patient setting. So
I know if your work ever strays into the diagnostic space, this is a group to talk with.
Yeah, exactly. Great. Thank you so much.
So take care, everyone, and we'll look forward to seeing you next week. And Helene, who's here with Mike Nimmo, will be presenting.
Thank you. Bye, everyone.
Yeah, bye-bye.
Have a great weekend.